Enzymes, their properties and examples. The functional enzyme is called as holoenzyme, and holoenzyme is made up of apoenzyme, which is the protein part of enzyme, and cofactor, which is the non-protein part of enzyme. So together, apoenzyme plus cofactor is called as holoenzyme. Now this cofactor, if it is inorganic or it is the metal ion, it is called as activator. And if the metal ion is tightly bound to the enzyme, then it is called as metalloenzyme. And if the enzyme is loosely bound to metal ion, it is called as metal activated enzymes. And if the cofactor is organic compound which is attached to apoenzyme, it is called as coenzyme. So coenzyme is an organic compound which is non-protein part, and it combines with apoenzyme to form functional enzyme, that is holoenzyme. Coenzyme is derived from vitamin B complex. There are some coenzymes which are derived from non-vitamins also. Some enzymes require both a coenzyme and one or more metal ions for its activity. When coenzyme or metal ion is very tightly or covalently bound to enzyme, it is called as prosthetic group. For example, the heme proteins like cytochrome oxidase, catalase, peroxidase, they have heme as a prosthetic group. Some authors use the terms like prosthetic group, cofactor, and coenzyme interchangeably. Now let's see the properties of coenzymes. So, as we know, holoenzyme is made up of apoenzyme plus cofactor, and this cofactor, if it is organic, it is called as coenzyme. So, apoenzyme is the protein part of holoenzyme, and it is responsible for three-dimensional infrastructure which is required for chemical reaction. And the coenzyme is non-protein part which is organic, and it is required for optimum activity of enzyme. And together, apoenzyme plus coenzyme, it is called as holoenzyme. So this coenzyme, it is low molecular weight organic substance. It is heat stable. It is the transient carrier of specific functional group, which is essential for biological activity of enzyme. And the coenzyme it combines loosely with the enzyme molecule, and it is easily separated by dialysis. So what is coenzyme? Coenzyme is a non-protein, low molecular weight, heat stable, organic molecule, dialyzable, and it is required for optimum activity of enzyme. After completion of reaction, the coenzyme is released from a coenzyme, and it can bind to another enzyme molecule. Most of the coenzymes are derived from vitamin B complex, and it has no role in determining substrate specificity of enzyme, because the apoenzyme, which is the protein part of holoenzyme, it is responsible for substrate specificity and not the coenzyme. Now let's see various coenzymes which are derived from vitamins, that is B complex vitamins. So B complex vitamins include vitamin B1, which is also called as thymine, and the coenzyme is thymine pyrophosphate, required for oxidative decarboxylation trans-ketolase reaction. B2 is riboflavin, and the coenzymes are FAD-FMN, which are required for oxidation reduction reactions. Vitamin B3 is niacin, and the coenzymes are NAD+, NADP+, and this is required in oxidation reduction reactions. Vitamin B5 is pantothenic acid, and the coenzyme is coenzyme A, and it is the acyl carrier. Vitamin B6 is pyridoxine, and the coenzyme is pyridoxal phosphate, which is required for transamination, deamination, and decarboxylation of amino acids. Vitamin B7 is biotin, and the coenzyme is biocytin. It is involved in the carboxylation reactions. Vitamin B9 is folic acid, and the coenzyme is tetrahydrofolate THF, and this THF is a carrier of one carbon group. Vitamin B12, it is called as cyanocobalamin, and it has two coenzymes, methylcobalamin, which is involved in the methyl group transfer, and deoxyadenosylcobalamin, which is involved in the isomerization. So these are the various coenzymes derived from vitamins. First, thymine pyrophosphate derived from thymine. FAD-FMN is in vitamin B6, vitamin B12. Dehydrogenase complex. Affected dehydrogenase NADP plus is the coenzyme of glucose 6-phosphate dehydrogenase. Coenzyme A phospholipid, that is uracil group, and this lipoid is required by from riboflavin. NAD plus NADP plus derived from niacin and coenzyme Q. Group 2 coenzyme pyrophosphate, which transfers hydroxyethyl group. Coenzyme A, which transfers acetyl group. Bio transfers phosphate group. The use of coenzyme in a reaction become critical and even more important than the use of substrate itself for normal body functions. And under such conditions, the coenzyme is called as co-substrate or second sub. Important than the change of substrate into process of glycolysis, glucose is converted to glyceraldehyde 3-phosphate through series of reactions. Glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate by the action of enzyme glyceraldehyde 3-phosphate dehydrogenase, and here NAD is reduced to NADH. The enzyme lactate dehydrogenase in anaerobic condition, muscle cramps and lactic acidosis, and it is harmful. But still it is formed because this is NAD formation is very important and it is required to continue the glycolysis under anaerobic condition. And this is because the glycolysis is the only energy producing pathway in the process of glycolysis in the anaerobic states as well as enzyme. In the enzyme is tightly bound to metal ions and that cannot be separated without breakdown and loss of enzyme activity. The examples are cytochrome oxidase, which enzyme is loosely bound to the metal ions and can be separated without breaking the enzyme protein, and amylase requires chloride. Enzymes are carbonic anhydrase, hexokinase, phosphofructokinase, enolase. Iron-containing metalloenzymes are cytochrome oxidase. Xanthine oxidase has molybdenum. This is very important for brief answer question. The question can be enumerate and coenzymes. The functional enzyme is called enzyme. So apoenzyme plus cofactor is holoenzyme. Cofactor can be activator or coenzyme. So the cofactor, the activator, if it is called as metal activated enzyme. Tetrahydrobiopterin. Coenzyme Q. Group 2 coenzymes are involved in the transfer of groups other than protons and electrons, transfer phosphate group. So this is all about